I'm very happy that to conclude our program today, we'll have Professor Ian Chubb, the Vice Chancellor of the Australian National University, and he will introduce the next speaker, Baroness Susan Greenfield. Thank you.
Colleagues, ladies and gentlemen, I've got a string of notes here, which I'll read out, boring as they are. But what I'll do is I'll editorialise a bit of colour into the notes as I go through them. Of course, Susan and I go back a long way to 1973, indeed, when she came into the laboratory that I'd just vacated in Oxford, and my ex-supervisor became her supervisor, and he was away a bit, so Susan and I did quite a few experiments together, went down the pub together a few times. That's all, I think, that we did together. And during the period from 1973 to 2010, we've kept in initially close but intermittent contact since then. So I know so much about her that I know what to leave out from an introduction like this so as not to embarrass her by revealing things that I do know. She was an undergraduate at Oxford University and took her DPhil in the Department of Pharmacology. That's where we met. That's where she persuaded me in 1973 to stop smoking, because when she smoked, I saw what it looked like, and I thought I should stop doing this to my image. But she came into the department in 1973 and worked through to a PhD. I think I'd left before you completed, as I recall, and but I did have some contributions to make, I suppose, in a sense, to editing the thesis and so on. So I know what work she did. She graduated, of course, in psychology, as is well known, and but wanted to get a little more science into her background and more science into what she studied, so therefore open up more opportunities for her, which is why she took a DPhil in pharmacology, which was essentially an experimental science department. She held research fellowships in the department, or has held, in the Department of Physiology at Oxford. That was essentially immediately postdoc, I think, as I recall. Collège de France in Paris, which was also pretty proximate to the DPhil, and the New York University Medical Center in New York. 1985, she was appointed University Lecturer in Synaptic Pharmacology and Fellow and Tutor in Medicine at Lincoln College in Oxford. And she became Professor of Synaptic Pharmacology in 1996 and has since been awarded 30 honorary degrees from British and foreign universities. 30 honorary degrees. I didn't know that they gave them away that easily, Susan, but... oh, really? Who? Flinders? It must have been after I left. Don't expect one here. No, there are mates' rates, come to think of it. So now that I know you're a wealthy woman. From 1998 until 2010, she was Director of the Royal Institution of Great Britain, and she held that post jointly with her chair in Oxford. She's currently Director of the Institute for the Future of the Mind at the James Martin 21st Century School, Senior Research Fellow at Lincoln College and Honorary Fellow at St Hilda's College, all at Oxford. Susan heads a multidisciplinary research group exploring novel brain mechanisms linked to neurodegeneration, and she also studies the physical basis of the mind. In 1995, she published her own theory of consciousness. In addition, she has developed an interest in the young mind and the impact of 21st century technologies on how young people think and feel. That'd be worth a lot to know that. In 2000, her research, even for Vice-Chancellors, when I think of the young people thinking and feeling that would be worth a lot, in 2000 her research was the subject of a chapter in Bold Science, a book profiling iconoclastic science pioneers. Susan has recently received the 2010 Australian Medical Research Society Medal. She was awarded a CBE in the Millennium New Year's Honours List, and in the same year was made an Honorary Fellow of the Royal College of Physicians. She was granted a non-political life peerage in 2001. She has been awarded l'Ordre National de Légion d'Honneur 2003 and was appointed Chancellor of Heriot-Watt University in 2005. Chancellors are very important positions in some institutions. In 2007, she addressed the European Parliament on the impact of technology, and in the same year was elected into the Fellowship of the Royal Society of Edinburgh. So Susan Greenfield will address us on the question, how does the brain generate consciousness? So ladies and gentlemen, it is a pleasure for me to invite my long-standing, I can't say old friend, long-standing friend, Susan Greenfield, to address us this afternoon.
Thank you. Oh, good evening. It always is a bit of a cliche to start off by saying it's a pleasure to be here, but it really is. It truly is a pleasure to be here for lots of reasons. First is to see Chubb again, who I haven't seen for all of 2 or 3 months. And it's true what he says. We do go back a long way, and I was sort of cringing there, just wondering what on earth he was going to say. So thank you very much for forbearing. I don't know what it's going to be like in the champagne afterwards, whether more stories will come out. But I was ready to reciprocate, thinking, well, I'm going on second, so whatever he says, I can match what he says. But luckily, I can hold that in reserve too. It's also, as always, a huge pleasure to be back here in Australia. The one thing that Chubb didn't mention, which I'm hugely proud of, is that in 2006, I was actually made honorary Australian for that year. And that doesn't mean to say I get to go through the customs any quicker or the immigration any quicker, but I do have a little plaque for that, and it always is something that I would do treasure as an accolade, because this does feel like very much my second home, having been Think in residence in Adelaide for two 2 or 3 month periods. Anyway, it's marvellous also to be here at this very important meeting and to actually explore and celebrate, if you like, biomedical research and its application to life and to enhancing the quality of life. I think it's very appropriate that I'm on last, because what I want to talk about really is something where you do need to sit back on your chairs. I don't think you're going to make any money out of it any day soon. But it does require, I think, very serious and deep thought, because it is perhaps one of the most exciting questions we can ask in neuroscience. I would say it was the most important question, or exciting question. It's also probably the one that we're least likely ever to be able to answer. Or may we? What I'd like to do with you this evening is just to show the contribution neuroscience might be able to make. In his very generous introduction, Julia said I was here to say how the brain generated consciousness. I think that you might want your money back if you think that that's exactly what I'm going to do. What I'd like to show, though, is perhaps how we can bring something to the party, because traditionally this is a question that's really been the province of philosophers. And what I'd like to do is show that neuroscience perhaps can make a contribution, or even if not, come up with the answer. Okay, so what's a great joy speaking now to an audience of card-carrying neuro and biomedico neuro types. I'm right in thinking that, aren't I? Everyone is pretty technical here. I've always... in which case, one can go fairly swiftly and actually share, I'm sure, the experience that most of you have had of holding a brain, rather as shown here, when you're doing an initial brain dissection. And I remember the first time I did that, thinking, if I got a bit under my fingernail, what would that be? What would that bit of tissue do under my fingernail? Would that be the bit that somebody loved with, or would it be a memory? But even more remarkable is to contemplate, as you hold, as I'm sure everyone in this room has done, as you hold the brain in your hand, to think, how do you get from this stuff which occupies the same universe as earwax and saliva and tooth enamel, how does it, unlike those other things, generate the experience that you're having right now? Well, I hope you're having it right now, unless you've fallen asleep. But that is to say, how does it translate? How does the water of the everyday machinations of chemicals and brain cells, how does it translate into this? This is George Bernard Shaw, who you may or may not be familiar with, but you will know, I'm sure, that he was a very eccentric and individual and highly original writer. But really, those writings were just a pale echo of what was going on behind those piercing eyes. And how do you get from the sludgy thing you can hold in one hand, get under your fingernail, how do you get from that to this? Or to you? I mean, however close you are to the people sitting next to you now, however articulate you are, or musical or poetic, they're not going to be able to understand what you're feeling firsthand. And it's this quintessential, subjective, first-hand experience that we informally call consciousness that really we're going to look at this evening. Now, already I've said something that's an anathema to science, and that is subjectivity. And that's why I think this is very much still an area that's shunned by certainly grant-giving bodies. You have to pretend you're working on anesthetics or schizophrenia or something. You can't actually apply, or perhaps you can, but in the UK, you wouldn't get a grant to study consciousness. Someone once described it as a career-limiting move, a CLM, if you were trying to do this. So it's something that many scientists are still very wary of, but as John Searle, the philosopher, says, as he said, to say that you're studying the brain but you're not interested in consciousness is really to say that you're studying the stomach but you're not interested in digestion. You know, it's what the brain does. So I'm very pleased to have the chance to share with you some thoughts, and welcome in the Q&A your own. I think, as all good scientists or any good student writing an essay will have to do, we have to start by being honest and making some assumptions. Otherwise, we're going to flounder for the whole evening just saying we can't define consciousness and just give up. So let's think about, therefore, the assumptions. I'd like to just put it to you because we are all scientists, and I doubt if there... Can I just check there's no philosophers here? Are there philosophers who dare put their hands up? Yeah. I'd like to say consciousness can't be formally defined, and that is because our two strategies for definitions don't work here. Either you normally refer to a higher set, so you'd say a table is a piece of furniture, love is an emotion, conscious is a what? What is it? It's not as if you can have a higher set. Or we have operational definitions. Love is when I give my mother 3 red roses. Flight is when you defy gravity. Conscious is when you do what? You're not doing anything now, but I hope you're, as I say, still conscious. Yeah? So given that those two strategies don't work, I'd like to just shelve the issue rather than give up on it, as some people do, that we can't define it. Because let me be very crude. We all know what consciousness is, don't we? It's what you're going to lose tonight. Yeah? So let's just assume that for the time being. And that brings us then to the hard problem, so-called by someone called David Chalmers, actually an Australian, who's earned his name and fame, I think from this university actually, by coining the phrase the hard problem, meaning nothing to do with attention or content, but the hard problem is what I've just said. What is the difference in consciousness and unconsciousness? What is the difference? What's happening in the brain between those two states? How does the water get turned into wine? That's the hard problem. So, what I'd like to suggest is that it's generated by far and of the brain. Again, this might seem obvious thing to say to neuroscientists, but there are so-called panpsychics out there, of which he is one actually, where the people actually talk about consciousness being an irreducible property of the universe, and your brains are kind of, satellite dishes that are receiving consciousness somehow. Now, although one can't refute that idea, you can't take it forward or do anything with it. So I would rather not dwell on it too much, but speak to you as neuroscientists and say, well, look, in my own view, and I'm sure you share this, if we are interested in consciousness, we're going to have to think of its provenance as being related to the brain. So finally, any so-called scientific, note the scare quotes explanation, unfortunately has to include subjectivity. We can't get out of that. And this is where we're having difficulties, and that's what I'd like to share with you this evening, of how we might turn something that's qualitative, something we hate. We hate that as scientists. We hate qualitative things. How can we turn it into something that is scientifically tractable? Okay, so let's look at some candidate approaches first of all, and perhaps an obvious one would be, especially nowadays as these images are getting more beautiful and more high-powered, would be brain imaging. Now, the problem with that, if you look at the brain here, is where would be the center for consciousness? I wonder. And what do we mean by a centre for consciousness? Yeah. In my own view, saying a centre for something is misleading because it suggests you know what's going on inside it. And it's really, as someone once said, it's an anaesthetic explanation rather than, or it's a description, it's not really an explanation. I'd like to suggest to you there isn't such a thing as a brain within a brain that is a centre for consciousness, because it doesn't make sense to have a centre for this or that that's unrelated to other parts of the brain, as if you have a series of independent mini-brains. You're not really solving the problem, you're just miniaturizing it. Just to illustrate that, some of you from the UK, perhaps who've lived there, may remember this comic called The Beezer, and these were the numbskulls And the numb cells, as you can see, were little men living inside this other man's head. And I think this is a very good analogy for the notion of centers for this or that, because here you have, I don't know, the nose cleaning department and the crisp eating department. And here you have the consciousness department. Now, if you said, okay, what's going on inside the numbskull's brain then? Are there mini numbskulls? And inside the mini numbskulls, are there micro numbskulls? And are there micro numbskulls? Are there nano numbskulls? You're not really solving the problem, are you? You're just pointing and trying to reduce it, but you're not actually saying how it is happening or why it's happening. So I think trying to posit that there is a center for consciousness doesn't really get you anywhere at all, and it doesn't make any logical sense. Moreover, it doesn't really work when you look at it empirically. This is some old work by a guy called Chris Pomfret from Manchester University, who looked at a series of human subjects whom he anesthetized with halothane and imaged their brains. And you can see here on the upper trace, multiple brain areas are active, and with the halothane, there's a uniform shutdown in all the areas. It's not as if there was one area that selectively was active and then was shut down selectively by anesthetic. So I think that if we are looking for, with brain imaging for areas, then that's not very helpful. Let's go to the other end of the scale, as I used to do, which is to look at single unit recording, as shown here. Now, can that help us? Certainly you have very high time resolution, much faster and a much better spatial resolution than with brain imaging. But how can we interpret what we see? My own view is now the problem is rather like this, and I don't know how many people recognize this individual. I should have Australianized it, perhaps had Ben Cousins. Is it Ben Cousins? Bill Cousins in? This is the guy I've been hearing a lot about, who retired yesterday. But the same idea, which is if you're watching a game of soccer, as you call it here, or football, and you had a video that only focused on one player, you couldn't extrapolate from that one player's behavior the progress and indeed the nature and result of the game. So looking at single recorded is a bit like that. Yes, you can learn a lot about one brain cell, but how it works in relation to the context of the function is really anybody's guess. So what we really need, don't we, is something in the middle. We need to link the macroscopic with the microscopic, and we would like ideally a high time and a high space resolution to do that. So what could we do? Well, let's look at a mid-level process, and one such was suggested by my old boss from NYU, Rodolfo Llinas, and I won't go into details here because I just want to use it to illustrate a point rather than to talk about his work. What he was suggesting, there was a series of complex interconnected loops between the thalamus and the cortex, generating the magic 40 Hertz oscillations, and that this, he suggested, was the selective pathway or system was a requirement for consciousness. I'd like to suggest, though, that necessity isn't sufficiency, and although if it's inactivated, you might deprive a brain of the ability to generate consciousness, that's not to say this is what it is all about. If you put this in a dish, you wouldn't have consciousness. Moreover, it doesn't explain many of the things we have to explain in a robust theory. For example, how anesthetics work, or how vision differs from hearing, or how, even, there may be this kind of thing. Where is the Rubicon, I wonder, where consciousness occurs and where it doesn't occur? As we all know, the brain has elaborated in evolutionary terms around the same theme. It's not as if we suddenly have a new bit of brain stuck on certain species who are conscious and others who are not. And I'll defy anyone to, with confidence, draw the line and tell me the anatomical, I say, Rubicon, above and beyond which you get consciousness and below which you do not. To emphasize that point further, let's go even lower or wider, if you like, in the animal kingdom. And I had a marine biologist work with me once, Toby Collins, and he pointed out this very interesting anomaly, certainly in the UK. The common octopus, and we all know now from the World Cup and Paul the octopus in Germany how clever they are, but this is a Paul-like creature who, because of their huge intelligence, even before Paul, we know that they can do memory tasks and so on, is under Home Office legislation. That is to say, you couldn't just take an octopus, well, not a common octopus, and chop its tentacles off and eat it. You couldn't do that, or experiment on it. However, the Elidiocehosa, a related species, very similar, is not covered by legislation, so you could do it to those. So that just shows you how arbitrary the line is between determining whether a brain is sensate or whether it's not. And I think that we have to be very careful where we draw the line. That's equally emphasized in this very charming picture, which I love, where this little dog here is not doing anything in the canine repertoire. Normally she would bite the lady, I imagine, or run away, or both, with the screechy violin. But I, for one, I'm sure you do, find it endearing because it is a human behavior, even though it's a dog that's doing it, and that's why we find it rather sweet. Now, this illustrates two things. One is, again, you can't infer inner states from outward behaviors, as shown here. And also, what do we do? How do we understand about animal consciousness? Are they conscious? Are they not conscious? Where do we draw the line? If they are conscious, are they really conscious like we are? No, not quite. But then on the other hand, the whole point of having pets is they seem to love you and like you and know you and have a relationship with you. So where are we? And how do we understand this? And that selective systems, I don't think, accounts for this conundrum. So let's think about that a bit more. And now I'm going to be slightly controversial, but I don't really want to get into the ethics of this. I just want to look at the science of it, which is whether a fetus is conscious or not. And this is a 7-week-old fetus, and it does beg the question. In the old days, people thought that the fetus was not conscious, that you were conscious at birth. But that means that there's something special about being born, either the manner of birth, as you're squidging down the birth canal. Well, if that's the trigger for consciousness, that's a bit tough if you're born by caesarean section. Or is it the time of birth, which means, I don't know if anyone here has had a premature baby. I'm sure you didn't think at 7 months, well, the baby's not 2 months yet before the baby's conscious, let's not bother going to see it, And then, at 9 months, magically all the lights go on and the baby's conscious. That's not quite right either. So how do we account for these riddles of animal consciousness or fetal consciousness in trying to understand how we can look in the brain, what kind of shopping list we can take to the brain. So the problem with selective systems, such as Professor R Llinas has put forward, I would say, is that there's a difference between necessity and sufficiency. Just because if you eliminate something, you eliminate the phenomena, does not mean to say that's all there is. Second, how would you explain the actions of anesthesia, which I think are very important, given that's one thing we do know, takes away consciousness. And another issue, which I find fascinating, we'll come to later, is the difference between hearing and vision. All of us here in this room know how vision and hearing are processed in the brain, and they're very, very similar, and yet the subjective experience of hearing versus vision is huge. And if we knew how that huge difference between the subjectivity of hearing and vision was achieved, given the relative comparisons with the objectivity of sensory processing, I think that would be very helpful, but that's not accounted for with those selective systems. And finally, non-human versus human consciousness and fetal consciousness. I'm not saying I can answer all these, but I think we need to be mindful of them, and I will try and do that with the theory that I'd like to put forward instead. The theory I'd like to put forward instead is prompted by a series of observation, and some of you may be familiar here with BIS monitoring, which was something developed in the States to get round the rare but very distressing occurrence of when a patient is awake during anesthesia and is paralyzed so they can't tell you they're awake, and indeed the equally rare but distressing condition of people dying through over-anesthesia. And this is a proprietorial method using the EEG, where, as you can see here, you get a number indicating the depth of anesthesia. Now, we've known, of course, that sleep, like anesthesia, comes in stages, and I was reflecting on this once while I was on holiday, and I thought, well, how about if unconsciousness comes in degrees, why couldn't consciousness come in degrees as well? Surely we are wrong to talk about consciousness versus unconsciousness. What if consciousness grew as brains grew? What if it was continuously variable? Now, this would be very helpful because it would explain the conundrum of animal and fetal consciousness, because we could say, yes, a rat is conscious, but not as conscious as a cat or dog. And a cat or dog is conscious, but not as conscious as a human. And a fetus is conscious, but not as conscious as a child, who's not as conscious as an adult. And what's even more interesting is you could then say, even as an adult, you will have varying amounts of consciousness. There can be times, perhaps, when you deepen your consciousness or raise your consciousness. In everyday folklore terms, we talk in this way, don't we? We seek out scenarios to raise our consciousness or deepen our consciousness. It doesn't matter which way we go, up or down, doesn't really matter. But we're talking now in terms of quantity. And here one seizes the chance, because once we talk about quantity rather than quality, the scientist has a chance. We have a chance now to look in the brain and say, is there something going on mid-level of organization between single brain cells, between macro brain regions? Is there something going on that's nice and fast that actually can vary from one moment to the next in degree? And I wouldn't have set up a rhetorical question like that if the answer was no. So what I'd like to suggest to you then is that we need to look for a mid-level analog process, something that varies, that's not just all or none, and that is continuously variable, not just across evolution and not just across development, but also within an adult individual brain. And what I'd like to suggest to you is that, and I'll use an analogy first, a metaphor, is that if we could find in the brain something that was like ripples. So what would happen is that you would have some kind of trigger for a moment of consciousness, something like a stone being thrown in a puddle. But that would generate, just for a moment, evanescent, highly transient ripples, the excursion of which would correspond to the degree of consciousness. I'll repeat that. What we're looking for is some mid-level phenomenon in the brain that only lasts for a fraction of a second, that is triggered by something, and that can vary in its excursions from one moment to the next, because those excursions will correlate, will match up with depth of consciousness at any one time. So let's see what there could be. Well, amazingly, when I visited the Weizmann Institute a while ago, I visited the lab of Amram Greenfield, and this is his work from a long time ago now. Now, he had been working with optical imaging, that is to say, fluorescent dyes. I don't know if anyone here works on these, but we do in my lab. I'll show you in a minute the results. And these dyes cannot be used on humans because they're potentially toxic, but they embed in the membrane and they change their wavelengths when the membrane depolarizes. So you get a very high space and time resolution readout of what the neurons are doing. And what he showed was, this is a flash of light to a cat. I hope you can see that I was delighted that it was like ripples. There was an exponential decay here coming from a epicenter, for want of a better word, and he reckoned this was about 10 million brain cells encompassed in the full excursion of these two ellipses. And moreover, the whole thing was corralled up a maximal at about a quarter of a second and then disbanded within about half a second. So this would never have been captured with contemporary fMRI or non-invasive imaging, and yet you see the brain is capable of doing this. The brain is capable of these very large-scale coalitions that are highly transient. So surely this would satisfy our requirement for that mid-level phenomena that we are seeking, and one indeed that could be variable. So I'd like to talk about these ripples in the brain then. And the word assemblies, again, we have to be careful with definitions because it's a term that's been used a lot in physiology and it means different things to different people. This is the particular definition I'd like to use this evening, which is that they are variable, highly transient, that's to say sub-second, macro-scale groups of brain cells, usually exceeding 10 million, that are not confined to nor defined by anatomical brain regions or systems. Okay, so this would be what we are studying and what we're going to manipulate to see if we can also manipulate consciousness. But just to show you one, here we have a parasagittal section from my student Michael Hill, and we're stimulating here in the thalamus and we're recording in the cortex, as you can see here, into area 17. And you can see here the time scale. It's 84 milliseconds in all, but it's obviously slowed down. So I'll just stimulate now, and you can see how it looks. So you're stimulating. So we know it's physiological because the stimulation is here, but the assembly is remote, as you can see, and then it goes down the cortex. It goes from area 18 to 18A. So that's what an assemblies look like, and that's the sort of working thing that we can study. This is rat brain in a dish. Okay, so if that is the case then, and we now have something to study, what would be the stone in physiological terms? What is the thing that triggers it? So let's have some thought about that. The obvious candidate is the brain cell. The single brain cell even looks a bit like a stone. But I don't know how many people here of the same vintage as me, apart from me and Chubb, who would remember the fallacy of the grandmother cell. And I always had problems anyway with this notion that you had one cell that was selective for one function finally, recognition of one face finally, because if you had a grandmother cell but you never had a grandmother, it'd be a waste of a cell. Or if that cell died, as many do, it means you'd never recognize your grandmother ever again, and that patently isn't the case either. So it didn't quite stack up to reality. I know the work of Christoph Koch, as some of you may, with his Halle-Berry neurons, and I have my own views on those, which we can talk about over the champagne if you wish. But suffice it to say, I think it is improbable that you have a single brain cell, the 100 billion cells you have, that corresponds to one thing, that if Granny comes into the room, is activating it, and that is like the stone being thrown in the puddle. Instead, of course, much in vogue are neuronal nets, and you can see this rather pretty picture here, where you have networks of neurons that would correspond to the stone. And I would like to suggest so-called hardwired groups of cells, a term, incidentally, I don't like because it invites, I think, misleading computer analogies, but it's one that's used a lot. Relatively hardwired brain cells forming a group that could be the stone. If that was the case, what would be the throwing of the stone? Well, it would be, in simple terms, just seeing something. Or it could be, for humans, something that wasn't purely sensory but cognitive. And as we all know, the wonderful thing about being born a human being is even though you're born with pretty much all the brain cells you'll have, it is the growth of the connections between the brain cells that accounts for the growth of the brain after birth. So even if you're a clone, an identical twin, you have this unique pattern of brain cell connections. And it is this unique configuration, the plasticity of the brain, that I like to call the mind. Now let's distinguish mind, brain, and consciousness. The brain is the sludgy physical thing you can get under your fingernails. The mind, I'd like to suggest, is the personalization of the brain, this long-term localized changes that we're all familiar with plasticity. Consciousness is often influenced by the mind, but it can be distinguished from it, because when you go to bed tonight, you're not going to lose your mind, are you? You're going to lose consciousness. Similarly, you can be conscious, but you can blow your mind or lose your mind. So I think we must differentiate the two. And again, I'll return to that in a minute. So here we are living our life in downtown Oxford. This is what it sort of looks like. Everyone's got their unique narrative, their unique time and space trajectory, and everything you do is going to leave its mark on your synapses. I don't need to rehearse too much the phenomenon of plasticity, but I just couldn't resist this one because it is very striking, of 3 groups of human volunteers, none of whom could play the piano. And if you ever get to do this, don't be the control group, because they just stared at a piano for 5 days. And if you look at the brain scans, you can see that over 5 days, very little happens to them. But the group that were asked to play the 5-finger piano exercises, there's an astonishing change in the functional error rate into the digits. But the most remarkable group, and that's why I included it, even for neuro experts such as yourselves, because I do find it fascinating, is that here, the group that just imagined they were playing the piano looked pretty much the same. So here it's the thought, as Hornikowitz said, the man who developed L-DOPA therapy, thinking is movement confined to the brain. Wonderful quote. And you can see here it's thinking. It's the thought that precedes the contraction of the muscle that is the all-important feature. So I don't need to dwell too much then on how we are familiar with how we locally, in inverted commas, hardwire our brains. We privilege certain connections, have certain personalized associations. Therefore, we shift from sensory to cognitive. And in so doing, I'd like to suggest we are increasing the size of the stone. E stands for epicenter here. So what we'd have then is either a sensory stimulus. The most simple one is, for example, an alarm clock. And I challenge anyone with rival theories of consciousness to explain why an alarm clock wakes you up, a very simple phenomenon you have to explain. Why does a loud noise bring you into consciousness, or indeed a very bright light bring you into consciousness, the daylight coming in? So that's purely sensory, or we can have a cognitive one. Here I like to show my colleagues. I don't know if Ian remembers any of these. I don't think so. They're deliberately shown because no one should be able to recognize them. They're just 6 white middle-aged men in suits and ties, my sort of typical colleagues. And I'm sure you could substitute similar ones. The whole point is that you will see them as generic people. I see them as people for whom I know their birthdays, the number of kids they've got, when their last paper was accepted, what grants they've got, what they want to do, what their hopes, fears are, what their reversals have been, and all about them. I know their names, everything, and just as you could. So for me, there's a very cognitive take on them. For you, it would be a more sensory one. And most of the things we have are a payoff or a mixture between the sensory and the cognitive. And depending on that, one could think of the stone in the puddle, the sensory being the degree of sensory stimulation, how loud the bell, how bright the light, and that played in with the other factor, which would be the extent of the associations, the cognitive element. So you can imagine a very large stone being thrown very forcefully, a little stone being thrown forcefully, a big stone being thrown gently, and so on, across that dimension. Suffice it to say, what will happen when you then see Granny or whoever, or hear the bell, that this will activate this local group of neurons, and we all know the mechanisms of that. It's long-term potentiation, long-term depression, but this is too local and it's too long-lasting to account for consciousness on its own. However, that will recruit, hopefully, the ripples. Now, how does it do that? Well, we need to think of another feature of the brain, and that is the fact that we have the old ascending reticular activating system. I'm sure many of you are familiar with that term. For people who are less familiar with that, that's, of course, the groups of the amines and acetylcholine, which are in the brainstem region, which have very global, diffuse projections throughout the upper regions of the brain and can indeed act like fountains. Now, these traditional transmitters, I'm sure you're familiar with, more recently, well, I say recently, over the last 10 or 20 years, have been studied also as modulators, where the chemical is predisposing the cell to respond in a certain way without itself eliciting the response, usually sensitizing the cell to a stimulation if and only if that stimulation then comes along, but it amplifies that stimulation. So this, it seems, this system could play a part in sensitizing or helping the stimulus recruit cells into the ripples. What you'd have is these cells, which vary with arousal levels and biorhythms, if you like, putting cells on red alert so that if they are active in conjunction with you seeing something, the two things combined would privilege or enable the recruitment, the wider recruitment of a transient group of cells into an assembly that wouldn't normally occur. So therefore they're quite important. This is simply an example of how even the most familiar of transmitters, acetylcholine, can act as a modulator, and they do it in different ways. For example, here the opening of potassium channels will hyperpolarize a cell. That could be seen as inhibition, but if it occurs following a stimulation, I don't know why that's gone to egg timer, with a stimulation, then you can actually deactivate a calcium conductance, which predisposes to bursts of action potentials. Or here you can close a potassium channel to stop the braking that occurs with trains, and therefore you'd again have a facilitation, even though you've got opposite actions on the channels. I'm sure many of you are familiar with the cleverness of different transmitters in conjunction with different receptors and ion channels, and the paradoxical results they can produce, so that there's no one transmitter having any one effect of inhibition or excitation. And we know that dopamine and so on can also all act in similar ways as modulators. So if that's the case, then let's put all this together and try and characterize how assemblies might be formed. We can look in coronal sections here at basic pharmacology, and you can see here how physiological indeed they are. This is the baseline effect here. Tetrodotoxin blocking sodium channels has stopped that effect, and here you can see there is a pharmacological dissection where it's sensitive to GABA B receptor blockade but not GABA A. And this is not really a highly technical talk, but I just wanted to put these slides in to show that we do have the ability now to actually do the nuts and bolts dissection, if dissections can be nuts and bolts, of actually trying to analyze the mechanics of what goes on with these assemblies. So let's now, though, turn to the more interesting functional applications. And I did touch at first on the difficulty of differentiating hearing from vision subjectively, when in terms of most physiology, the two are not that different. However, if you look at assemblies, you do see a very dramatic difference after 300 milliseconds. And you can see here in 11 different experiments how the epicenter for the visual system ends up quite deep into the cortex, whereas for the auditory system, the reverse is the case. Now, how that maps onto the phenomenology, we don't know, because the issue is not so much differentiating the physiology, what we see, but how do we map this on to hearing and vision, which are, in phenomenological terms, so very different from each other. We need a common yardstick for describing the two, and the nearest thing I can come up with is actually in terms of time-space, where we could say that hearing is primarily but not exclusively a difference in time, whereas vision is primarily but not exclusively a difference in space, and the two could be along the same space-time continuum. But given I'm not an expert on relativity, I would flounder at that, but I think there may be a future for modeling these two modalities along that same dimension, and then mapping that onto this perhaps in the future, and it's something one could do in the future. Suffice it to say, though, that a slice is never conscious, and these assemblies, if they're going to be useful for studying and looking at ripples in the brain, we need really to turn to in vivo. With humans, as we've seen, the time resolution and space resolution of non-invasive imaging is very restrictive, but I think one could start to extrapolate, because you can do both in a rat, and at the same time you can do the invasive technology in a rat and try and see how it marries up. So this is in vivo now, and this is an in vivo clip. You can see it's 30 milliseconds normally, and you can see that compared to the fMRI, we are at a very different time and space scale. So it would be quite interesting to look in vivo, and you can see here that with anesthetic, as one would have predicted, as you deepen the anesthesia, so you do indeed reduce the size of the assembly. Other things that we can do is, for example, look at chronic effects, and one of the paradigms that we've studied is an enriched environment, and you can see here the rat's looking quite happy in this very stimulating environment, and I know that this was pioneered in Melbourne with the Huntington's chorea work, and that's been very impressive. So what we've done here is to look at assemblies where we know that the effects can be an increase in dendrite growth. What we've looked at is assemblies, and behold, we do indeed see that the enriched condition in the assemblies are bigger in animals, and this is now looking at them in vivo. So if enrichment enhanced plasticity increases assembly size, could the reverse occur? And could we have that if you have enhanced assemblies, I wonder if you would then have an increase in plasticity, and that's something that we'd like to explore. If that is the case, and this is just a speculation, then that might tell you why the assemblies have an evolutionary value, because it would facilitate the coordinated and global adaptation to the environment, because they are very global in the way they are spreading out throughout the brain. And therefore, the more adaptive the species, the larger the assemblies would be. And what that would mean, if this is the case, that perhaps if the assemblies are correlated with consciousness, then perhaps that's the function of consciousness. It's a corollary of assembly formation, and assembly formation is there to facilitate global adaptation in the brain to changes. So that would be one thought. So neural assemblies then, as I said, I can't come up with all the answers, but they are brave attempts to try and get around the issues that the selective systems could not, of explaining non-human versus human consciousness, the BIS monitoring, the efficacy of the alarm clock, hearing versus vision, anesthetics, evolutionary value. Let's now turn briefly to the theoretical applications. Can we, if this is true, can we now have a bilingual description of consciousness? Because although we can't turn the water into wine, can we go from physiology to phenomenology and then back again? Any good correlation, you should be able to do that. So let's start first with physiology. What kind of consciousness might we have if we had an abnormally small assembly? I wonder if we could predict what kind of consciousness we might have. Now, the beauty of this model, she says modestly, is that there's different ways and reasons for having a small assembly. So let's see what they are and see if the kind of phenomenology is common to all these different states. First is childhood, as we've seen already, modest connectivity. The second would be dreaming, where you have a weak sensory stimulation. So if you didn't, you'd be awake. It's dependent on the residual electrical discharge within the brain. Schizophrenia, where there is abnormal modulation of particularly the dopamine, but other amines in those fountains. Fast-paced sports, where you have competing assemblies, possibly, because so much is coming thick and fast. And finally, raves, or whatever they're called nowadays, where people take drugs to impair the functioning of their synapses, and I would argue it's a world stripped of all cognitive content. So we go techno. So if that's the case, do any of these conditions have something that could be in common if they're all small assemblies? Well, let's think about the rays. Back in ancient Greece, people talked about having a sensational time. I don't think anyone would come with me now if I said, let's go out now and have a cognitive time. It's very interesting that letting yourself go, blowing your mind, losing your mind, the very word ecstasy in Greek is to stand outside of yourself. It's very interesting, this abrogation of the sense of self. Very interesting idea. And this I couldn't resist. This is actually from Queensland. I know there's one in the audience from Queensland. I love this one for hovercraft fun rides. Totally out of control is a selling point. It's quite interesting that we pay for money to be out of control. So in all these cases, these people are in the moment. They are not in a world that's highly cognitive. There's no past or future. And this is the sort of state of mind that, if you like, we could call highly aroused, addictive, or rewarding, and all of these, in turn, have in common a particular transmitter, not the only one, of course, dopamine. So, how does this fit in? Well, we know dopamine is one of the agents in the base of the brain, and with schizophrenia, of course, we have inhibition of the frontal lobes, and it's in excess. And so can we distinguish two basic modes of the human brain, one of which would be like the small assemblies, with prefrontal underfunction as opposed to activation? The prefrontal function, the small mode assembly, if you like, would be one of strong feelings. The prefrontal normal thinking dominates, sensory versus cognitive, if you like. One characterized, as we've just seen from those little shots, from the here and now. The other, a narrative of past, present, and future. One, the external environment is driving and dominating, like with a small child, ice cream, you drop the ice cream on the floor and you say, look at the bird, it looks up at the bird. You can't do that with a depressed adult. Internal perceptions dominate. This has little meaning. What you see is what you get. Here is a highly personalized cognitive meaning, like my colleagues at the work. Here there's a reduced sense of self. You've let yourself go, after all. Here a strong sense and a continuity of self. Here no time or space. Here a clear episode. This, infants and children mainly, and animals. Here older children and adults. Here I'd suggest more dopamine. Here less dopamine. So if that's the case, and we look at the prefrontal cortex, as you know, that facilitates cohesion with the rest of the brain. It has more outputs than any other of the cortex. So could this hyperactive PFC, prefrontal cortex, could that be a factor in the small assembly mode? Here we have our friends at the disco. I would suggest that they are stripped of all cognitive content. They're in a world that's a booming, buzzing confusion, dominated by the here and now senses. If that is the case, if that's the small assembly, therefore, what would be the opposite of a large assembly profile? What would be a world that was remote? It'd be a world where the senses this time were understimulated. there'd be a strong continuity of thought. And that, I'm nervous to talk to an expert on depression here, but that would be perhaps clinical depression, where there is again an imbalance of those fountains, hence the efficacy of Prozac, characterized by anhedonia, a lack of pleasure, a lack of doing the kind of things we've seen, and above all, a numbness of emotion, as opposed to the strong emotion. I love this picture of Fragonard's swing. People are abandoned, they're letting themselves go. So could it be, if we put those two things together, that the greater the neuronal assembly, the less emotion, and therefore, perhaps not surprisingly, emotions as the most basic form of consciousness. You only have to think about the tail-wagging dog or the purring cat, the gurgling baby, I think, to intuitively feel that that has to be right. So that would be the small assembly world and the large assembly world. Can we go the other way? And can we take a phenomena that we've all know, let's take pain, for example, and explain that in terms of assemblies? Neuropathic pain, I'd like to suggest, is characterized by the size of neuronal assembly for the following reasons. First, we know that pain is expressed in other associations, pricking, stabbing, burning. Second, we know there's a diurnal threshold to pain. We know these rather macabre experiments on poor students where they had electric shocks through their teeth. And there's a change in the threshold to pain throughout the day, and that's not due to change in the conduction velocity of the pain fibers, that's due to changes in the modulation within the brain. We know greater anticipation, the greater the pain, you're building up an assembly. We know that phantom limb pain, which Mark Moore called the neuronal matrix, I would call an assembly. If that's activated, that can cause, if it's inappropriate, proprioceptive feedback, phantom limb pain. We know that pain is absent in dreams, which I've suggested is a small assembly state. We know that morphine, obviously an analgesic, gives a dream-like euphoria by working at the enkephalin receptors, and I'd suggest, therefore, reducing the size of the assembly. People say that with morphine, the pain no longer matters. They feel it, but it is no longer significant anymore. And in schizophrenia, rather horrible experiments to do someone who's already quite tortured mentally, people have found that if you give pain to a schizophrenic, they have a higher pain threshold, whereas conversely, to people who are depressed, they have a lower pain threshold. There's a comorbidity, as I'm sure you know, for hyperalgesia and depression. And finally, we know that anesthetic action could be explicable in this way because it could be shrinking the size of the assembly. And if that were the case, you'd say, well, as you're undergoing the anesthetic, you'll go through that pleasurable small assembly mode, which sounds a bit strange, and of course doesn't happen nowadays because of the efficacy of anesthetics. But I will remind you, in the old days, people were given ether frolics, and they were given ketamine, low-dose ketamine, for pleasure, and you'd have it at fun fairs and so on, low doses anesthetics to give you a high. Okay, so what would the future hold then if this model can link phenomenology and physiology backwards and forwards? Well, I'd like to suggest to you that we can be bilingual indeed. Degree of neuronal activity, the physiology, could be talked about phenomenologically as intensity of the senses. The extent of pre-existing associations, as we've seen, could be the significance to you personally. The availability of modulators, such as dopamine and other transmitters, could be arousal. The availability of other modulators, which I haven't mentioned, such as hormones, could be predisposition or mood. The formation of the competing assemblies would be a distraction, and input from prefrontal cortex would be your life narrative. So one can actually start to see how it's bilingual, and you can, I'm not going to go through this in detail, on the one hand have the physiology with the phenomenological counterpart on the bottom, and make predictions as to the size of assembly and turnover and so on. Now, I don't care if this is right at all. That's not important. What does matter is that it's testable. And that's why I think it's important that we can at last use science to develop falsifiable hypotheses. But we mustn't forget that the brain is embedded in the body, and it's not a disembodied brain in a vat, but somehow we have to think about how the brain works with the rest of the body. So I'd like to conclude by doing that. What I'm suggesting is that you do have a hub of cells hardwired, and they are stimulated to varying degrees, determined by the extent of the cognitive associations, modulated by various agents coming out from the brainstem and other hormones, plus the formation of competing assemblies, all of which will determine the final size of a neuronal assembly at any one stage. But then that is not enough. This is an index of consciousness, not consciousness. And my suggestion is that you'll have signature agents, and I think peptides, my money goes on the peptides, because they can be trilingual with the other control systems of the body, the immune system and the endocrine system, and also because we know they are co-stored with transmitters and released under very selective and special and different types of events, which means that they can be qualitative almost in what they are reflecting. That would then contribute to the circulation, feedback to the rest, and so this iteration of peptides as released as a result from the brain, the size of the assembly and type of assembly, and then being influenced back by peptides coming in from the body. It is this iteration that I would call consciousness. So you do need the brain and the body. This is all a bit garbled, and so this is a shameless plug for my book, which says it all a little bit more calmly. But I'd like to end by perhaps the most important slide of all, which I did say I wouldn't be able to deliver, which is it's all very well to start off with correlations like the one I've attempted, but do remember this. Here we have, say, phenomenology on one side and physiology on the other, and at the moment it's still at this stage. We do not know how the miracle happens for turning the water into wine. Thank you very much.
So, colleagues, time for questions, and catch my eye before you shout, and Susan will tell us a story. Who's got one? Yep, Jill. Yeah.
Okay, so we know from very recent work, and there's this astonishing tennis playing test that made people review how they see people who previously they thought weren't necessarily conscious. The work of Plum and Fogg from a long time ago has shown that what seems to be the case is that you have islands of activity rather than a cohesive large scale. So it does beg the question, how big an island do you need in order to have an inner state of consciousness? And I think one can't eliminate, and it may even be very possible, that there is some type of consciousness on the scale. If you buy into the idea that consciousness is a continuum, then it would be quite a straightforward concept that there is somewhere along that continuum a degree of consciousness. Now, whether it's a nightmare or whether it's a pleasant state, of course, is much harder. I don't know whether one could look at, galvanic skin responses or these sorts of things to see if people were undergoing a stressful type of experience. But I think it's always best to assume that something is the case rather than it isn't, and that's why I for one, have a problem with fetal consciousness when people assume that it's not the case.
Julia.
Hi, first, thank you for a wonderful presentation, and I'd like to ask you about this issue of dopamine and depression, which I think this whole aspect of what you presented related to depression is very understudied, and people look at other things and not so much at the reward circuitry and the role of dopamine. Do you have some comments about that?
Yeah, well, I think that dopamine, along with other chemicals, what I find very interesting is what they do to the holistic organization of the brain. I get very fed up, as I'm sure many people here do, as perhaps you do, reading that dopamine is the molecule for reward, as though it's got reward trapped inside it. Yeah? But what I find very interesting is the modulatory ability of dopamine, and indeed other agents, and you can see how versatile they are from even the two little slides I show with acetylcholine. They are so versatile. And dopamine, what I find very exciting, is according to the amount of dopamine, you could have opposite effects. Yeah? So if you had, say it was opening potassium channels, if you had it very inhibited, very hyperpolarized, you'd have inhibition. But if you had it slightly less hyperpolarized, but still via the same mechanism, you could activate calcium conductances. So you can have opposite effects. And I think that we have to be a little bit more sophisticated in how we're evaluating, first of all, medication that manipulates those systems in terms of the actual local concentration they're having and the cells and the ion channel profiles that are there, because you can have very different effects within that group. Yeah.
Yeah.
Do you think that it's possible to have multiple consciousnesses at a single time in a single brain? I'm probably thinking about split brain individuals, but it doesn't have to be the case. Could we have multiple consciousness that occur at the same time and
Okay, so it's like, okay, let me try and unpack that because it's an interesting question. And some people, and I don't agree with them, like Christoph Koch, talk about visual consciousness as though it's one type of consciousness, and say auditory consciousness will be something else. And I think what he's meaning is not what I mean by consciousness, because my own view is that consciousness is holistic, but very complex. But very complex. There's lots of things going on within it. So if you're asking, are there many elements to consciousness, then yes. But my own view is that they are normally working together in a kind of orchestra, rather than being separate, because I don't know of anyone who'd say that they had two consciousnesses going on at once that were completely separate. You have a complex consciousness.
So how do you resolve studies that have shown in split brain patients that if you phrase a question, you say that you show the same stimuli, and you phrase a question that is going to speak more to the left or the right brain, and you use the appropriate responses, that you get different results, possibly at the same time. Or even patients who know that they can see that his leg is...
Sure. Okay. Yeah. Okay. Yeah. Okay. Let's take the split brain first. I agree that you get different responses according to which hemisphere you present to, but I've always had a problem with that because the anterior commissure is still intact. Yeah. So and also we know that the rest of the brain's intact, so it's only a certain part of the connections between the two hemispheres that's severed. So we do know that you are dealing with a brain that is not completely bifurcated, in any event. And I think the person doesn't say they're two people ever, or they may have different responses to these two things. And then the other case was, sorry, what was the other one? It was the
foreign limbs syndrome.
Well, I think that might be when they neglect or they say it's, they're still them. I think that what we have to distinguish is content of consciousness from consciousness. Yeah. So these are very interesting cases of content of consciousness, but it's a bit like with blind sight, if any of you know that the person is conscious throughout that experiment. Yeah? So what you're manipulating is not consciousness, you're manipulating content of consciousness. Yeah? And I think content of consciousness is a separate issue, and that's what throws up all those very interesting things. But consciousness can only be manipulated by taking it away or not, yeah? Or by different degrees, I think.
Okay, yes.
Would you like to comment on the size of these neuronal assemblies, contrast between selective attention, you're focusing on one small thing, and the creative experience, where your focus has dissipated, like you've got a torch on a piece of paper, and when you're focusing on something, it's a very small circle, when you draw the torch, it spreads. Is that, in your opinion, what's going on during creative experiences? You've got a very large...
Yeah, with creative experiences, I think we could do it like that a little bit. And I think there's 3 stages to the creative process, yeah? The first is you have to deconstruct. So you have to challenge dogma. Now, whether that's in science, as it would be for most of us, to say, but why are we assuming such and such, yeah? Or with painting, where you have to deconstruct to shadows and lines and colours and textures, opposed to seeing an object. So I think in everything that is creative, there's a certain amount of deconstruction first, and one can imagine a sort of breaking of various associations. I think the next element is bringing together novel associations, but then you might say, well, look, every schizophrenic, just because they've got new words, is not necessarily, despite what R.D. Laing said, they're not necessarily brilliant poets, yeah? And people can be poets without being schizophrenic, so there's a sort of disconnect there. So the second element is, yes, you need unusual things coming together, but that's not good enough to justify it being a work of art or whatever. Yeah, I could come up with some science thing, all of us could, some experiment that brings two things together, but it's not necessarily great science. The brilliant thing, and the hardest thing, is the third, which is it has to have a meaning. So you have a deconstruction, then you have two new things together, but that must trigger vast associations, because meaning for me is seeing one thing in terms of something else. If you can do that, then you say aha or eureka, or the person that's watching it. Yeah? And I'm a great admirer of Baroness, the Australian immunologist who brought the principles of Darwinian evolution into the immune system. An astonishing link between two completely disparate worlds. Yeah? Really, now that, you say aha or eureka, and I can see some people nodding, that was a really remarkable intellectual leap, creative leap. Yeah? So the more you can do it across wider disciplines, the more fantastic it is. So yes, in one sense, I can do it in terms of associations and making a bigger assembly where suddenly two things have come together, or you can see one thing in terms of something else you never saw before. So it helps you, or indeed the reader or the observer or whatever, consumer, to say, now I understand, now I see it in a different way. Very exciting.
Yes.
Somewhere embedded in this is the attending mind, like the famous things in the psychology book about the mother who sleeps through a truck coming into a front lounge room but wakes up when the baby cries. And then there's a doctor waking up straight away when the phone goes but sleeping through the baby crying. Where does the attending mind come in? Is that just set the size of the stone?
Yeah, I think that is the associations. We all know that you can take something rather bland or neutral, but if it has a significance to you, it will be invested with more associations. So, for example, usually the numbers on your safe or your security numbers may not mean much to anyone. They might just be numbers, but to you they're your birthday, your wife's birthday, or something like that. So the more something can have a personalized association to you, in neuroscience terms, I would interpret that as having spawned, because it's special to you as you are living your life, more and more associations or connections. So that will mean it's much more readily recruited than it would be if it didn't have a large networks observing it.
Clinically, that has more of an adrenergic flavour now currently, doesn't it?
Yeah, I think that these amines, I tend to be rather generalized with them. You know, dopamine, noradrenaline, serotonin, we know that they are all very close, you know, where they are, and acetylcholine, and they all seem to spray out in this different way. I know one can differentiate them slightly, but I wouldn't say that one transmitter did one thing, one did something else. I think they can, in very subtle and different ways, act as modulators. Yeah.
Last question.
I was just wondering, this might be another one of the contents of consciousness, but do you have like an idea about people consciously deciding to do things? So like, whether that's a stone, you know, is that
Sure.
stones are sensory from outside, but people that sort of
Yeah.
can you generate things?
Okay. So this is something you ought to have another conference on, which is free will, yeah, which is a fascinating subject. And I don't know, I'm sure most people will know the Libet experiment. You know, Benjamin Libet, you know this experiment? Well, it basically asked the subject to press a button whenever they wished, and the EEG had changed already beforehand. And I myself have done that. I've been a subject actually on telly, looking rather stupid with the EEG cap on, you know. And it is very, very disconcerting, because you press the button when you think you want to, and already your EEG profile has changed, yeah? But again, as John Searle, who's my favourite philosopher, he said, look, when I go into a restaurant and I order a hamburger, it's because I want a hamburger. It's not because my brainwaves have changed or my genes are telling me anything. So I want a hamburger. So he argues, if you have the illusion or the subjective impression of free will, then by definition, you have free will. Now, of course, you can deconstruct it, but what you have to live with, and this is why it's a rather weird thing, is the physiology and the phenomenology as together and equally valid. Yeah? It's a little bit like quantum theory, where you have position and momentum, but you can't have both. You know, both are equally valid as explanations, but they have to live together, and you either have one or the other. You can't have both at once, and it's a bit like that. You know, you have to think of free will as, yes, I'm experiencing free will now. I'm asserting I want the hamburger. At the same time, yes, I can see if I'm looking at the brain, there have been changes that precede that, but there's certainly no little man. And this is what got the whole TV crew really kind of arguing, saying, but I'm not going to decide to do this. I know what my brain wants to do. And I say, you are your brain. That's all you are, of course. Yeah. But it is a rather thing. I said, the interesting thing is quantum theory, thinking of the two different modes of position and momentum. You have to think of the two as living together, but never actually converging. Yeah?
So the thing to take away is you are your brain, that's what you are. So just imagine yourselves as, I think what you described as a splodgy thing you can catch under your fingernail. And Julio's got something for you.
Thank you. Thank you.
So I'd like to make a final question, which doesn't have to be answered. It's more of a rhetorical question, which is that if you have the illusion of being rich, are you rich? And I'd like to present you with a moment of a Canberra consciousness frozen in time by a local artist.
Thank you very much.
Okay.
Thank you. Thank you very much.